Recombinant DNA. What would you change in your DNA code? So you want to transfer your DNA and change it. The first step is to make a copy of your DNA. The first step in using the polymerase chain reaction method to copy a gene is to heat a piece of DNA, which separates its two strands. Then, as DNA cools, primers bind to the single strand. Next, DNA polymerase starts copying the region between the primers. These copies can serve as templates to make still more copies. Until recently, plant and animal breeders could only work with variations that already existed in nature, and the changes produced by mutations were unpredictable. Today, genetic engineers can transfer certain genes from one organism to another, designing new living things to meet specific needs. It is relatively easy to extract DNA from cells and tissues. The extracted DNA can be cut into fragments or manageable sizes using restriction enzymes. These restriction fragments can then be separated according to the size using gel electrophoresis or another similar technique. In gel electrophoresis, DNA is broken into parts and a mixture of DNA fragments are put here in the gel. There's a power source, and the longer fragments will not travel as far, but shorter fragments will travel further, separating DNA portions. So in 1987, Douglas Prusher, a biologist in Woods Hole Oceanographic Institute in Massachusetts, wanted to find a specific gene in jellyfish that codes for a molecule called green fluorescent protein, GFP. This natural protein found in jellyfish absorbs energy from light and makes parts of the jellyfish glow. Prusher thought that GFP from the jellyfish could be linked to a protein when it was being made in a cell, a bit like attaching a light bulb to the molecule. To find the GFP gene, Prusher compared parts of the amino sequence of GFP protein to genetic code table and was able to predict the probability of mRNA-based sequence that would code for this sequence of amino acids. Next, Prusher used a complementary base sequence to attract an mRNA that matched his prediction and would bind to that sequence by base pairing. After screening a genetic library with thousands of different mRNA sequences from jellyfish, he found one that bound perfectly. To find the actual gene that produced GFP, Prusher took a gel in which restricted fragments from the jellyfish genome had been separated and found out that one of the fragments bound tightly to the mRNA. That fragment contained the actual gene GFP. This method is called Southern blotting, after its inventor, Edwin Southern. Gel electrophoresis separates DNA fragments produced by restriction enzymes. DNA cut with restriction enzymes. Bands of the gel are immobilized by blotting into nitrocellulose paper. Nitrocellulose paper, filter paper, an alkaline solution, and radioactive probes bind the fragments with complementary base sequence, the probes, and labeled bands. And this is the autoradiograph. Today, it is often quicker and less expensive for scientists to search for genes in computer databases where the complete genome of many organisms are now available. Are we developing into the database society? Once biologists find a gene, a technique known as polymerase chain reaction, PCR, allows them to make many copies of it. A piece of DNA is heated, which separates into two strands. DNA fragment to be copied. At each end of the original piece of DNA, a biologist adds a short piece of DNA that complements a portion of the sequence. These short pieces are known as primers because they prepare or prime a place for DNA polymerase to start working. DNA polymerase copies the region between the primers. These copies then serve as templates to make more copies. DNA polymerase is working in two directions. In this way, just a few dozen cycles of replication can produce billions of copies of DNA between the primers. What would you do with the recombinant DNA? Recombinant DNA technology, joining together DNA from two or more sources, makes it possible to change the genetic composition of living organisms. Scientists noted that Frederick Griffith's bacterial transformation experiments had changed the DNA of a living cell. During transformation, a cell takes in DNA from outside the cell, and that added DNA becomes a component of the cell's own genome. Griffith's extraction of heat-killed bacteria contained DNA fragments, which were taken up by the live bacteria, transforming the live bacteria and changing their characteristics. Scientists began wondering if it might be possible to change the DNA of a living cell, and many of them realized that this had already been accomplished decades earlier in Frederick Griffith's bacterial transformation experiments. During transformation, a cell takes in DNA from outside the cell, and that added DNA becomes a component of the cell's own genome. Griffith's extraction of heat-killed bacteria contained DNA fragments, which were taken up by the live bacteria, transforming the live bacteria and changing their characteristics. Today, scientists can produce custom-built DNA molecules in a lab and then insert these molecules, along with the genes they carry, into living cells. Machines known as DNA synthesizers are used to produce short pieces of DNA up to several hundred bases in length. These synthetic sequences can then be joined to natural sequences using DNA ligase or other enzymes that splice DNA together. A gene from one organism can be attached to the DNA of another organism. Restriction enzymes cut DNA as specific sequences, producing sticky ends, which are single-stranded overhangs of DNA. Restriction enzyme cuts the DNA, fragments join at the sticky end. Here's the sticky end, and the DNA ligase make the recombinant DNA. If two DNA molecules are cut with the same restriction enzyme, their sticky ends will bond to a DNA fragment that has the complementary base sequence. DNA ligase then joins the two fragments. The resulting molecules are called recombinant DNA. Splice it, open, add a piece of DNA, and it becomes a recombinant DNA chain. Recombinant DNA technology, joining together DNA from two or more sources, makes it possible to change the genetic composition of living organisms. By manipulating DNA in this way, scientists can investigate the structure and functions of genes. Scientists working with recombinant DNA soon discovered that many of the DNA molecules they tried to insert into host cells simply vanished because the cells often did not copy or replicate the added DNA. Today, scientists join recombinant DNA to another piece of DNA containing a replication start signal. This way, whenever the cells copy its own DNA, it copies the recombinant DNA also. In addition to their own large chromosomes, some bacteria contain small circular DNA molecules called plasmids. Joining DNA to a plasmid and then using the recombinant plasmid to transform bacteria results in the replication of the newly added DNA along with the rest of the cell's genome. Plasmids used for genetic engineering typically contain a replication start signal called the origin of replication, ORI, and a restriction enzyme cutting site such as EcoR ligase. Plasmids are also found in yeasts, which are single-celled eukaryotes that can be transformed with recombinant DNA as well. Biologists working with yeast can construct artificial chromosomes containing centromeres, telomeres, and replication start sites. These artificial chromosomes greatly simplify the process of introducing recombinant DNA into the yeast genome. Bacteria can be transformed using recombinant plasmids. Scientists can insert a piece of DNA into a plasmid if both the plasmid and the target DNA have been cut by the same restriction enzymes to create sticky ends. So plasmid DNA transformation using human growth hormone. So the human cell, this is the gene for human growth hormone. And here is a bacterial cell, bacterial chromosome, and here's the plasmid. And here's the connection. Opens this, and the sticky ends combine because they match, and the DNA recombination is inserted. So this is the recombinant DNA inserting this human gene growth hormone into the plasmid of the bacteria cell. And then that is placed back into the bacteria, the DNA insertion, bacterial cell combining gene for human growth hormone. As we showed, the new combination of gene is then returned to a bacterial cell, which replicates the recombinant DNA over and over again and produces human growth hormone. So that recombinant DNA, our purpose was to recreate this gene for human growth hormone. So the bacterial cell contains that gene and will reproduce itself with that gene. The recombinant plasmid has a genetic marker, such as a gene for antibiotic resistance. A genetic marker is a gene that makes it possible to distinguish bacteria that carry the plasmid from those that don't. This plasmid contains the antibiotic resistant gene TET-R or AMP-R, TET-R or AMP-R. After transformation, the bacteria culture is treated with antibiotic. Only those cells that have been transformed survive because only they carry the resistance gene. How can genes from one organism be inserted into another organism? Transgenic organisms can be produced by the insertion of recombinant DNA into genome of a host organism. The universal nature of genetic code makes it possible to construct organisms that are transgenic, containing genes from other species. Transgenic organisms can be produced by the insertion of recombinant DNA into the genome of a host organism. Like bacterial plasmids, the DNA molecules used for transformation of plants and animal cells contain genetic markers that help scientists identify which cells have been transformed. Transgenic technology has improved using mice in the 1980s. Genetic engineering can now produce transgenic plants, animals, and microorganisms. By examining the traits of genetically modified organisms, it is possible to learn about the function of the transferred gene. Many plant cells can transform using agrobacterium. In nature, this bacterium inserts a small DNA plasmid that produces tumors in plant cells. Scientists can deactivate the plasmid tumor-producing gene and replace it with a piece of recombinant DNA. The recombinant plasmid can then be used to infect and transform plant cells. The transformed plant cells can be cultured to produce adult plants. Transforming a plant with agrobacterium. The gene should be transferred. This is the recombinant plasmid is put in the agrobacterium. Transformed bacterium introduce plasmids into plant cells. Inside the plant cells, agrobacterium inserts part of its DNA into the host cell organism's transferred gene. A complete plant is generated from the transformed cell. There are other ways to produce transgenic plants as well. When their cell walls are removed, plant cells in culture will sometimes take up DNA on their own. DNA can also be injected directly into some cells. If transformation is successful, the recombinant DNA is integrated into one of the plant cell's chromosomes. Scientists can transform animal cells using some of the same techniques used for plant cells. The egg cell of many animals are large enough that DNA can be injected directly into the nucleus. Once the DNA is in the nucleus, enzymes that are normally responsible for DNA repair and recombination may help insert the foreign DNA into the chromosomes of the injected cell. Recently, it has become possible to eliminate particular genes by reconstructing DNA molecules with two ends that will sometimes recombine with specific sequences in the host chromosome. Once they recombine, the host gene normally found between those two sequences may be lost or specifically replaced with the new gene. This kind of gene replacement has made it possible to pinpoint the specific functions of genes in many organisms, including mice. A clone is a member of a population of genetically identical cells produced from a single cell. The technique of cloning uses a single cell from an adult organism to grow an entirely new individual that is genetically identical to the organism from which the cell was taken. Clones of animals were first produced in 1952 using amphibian tadpoles. In 1997, Scottish scientist Ian Wilmot announced that he had produced a sheep called Dolly by cloning. Animal cloning uses a procedure called nuclear transplantation. The process combines an egg cell with a donor nucleus to produce an embryo. First, the nucleus of an unfertilized egg cell is removed. Next, the egg cell is fused with a donor cell that contains a nucleus taken from an adult. The resulting diploid egg develops into an embryo, which is then implanted in the uterine wall of a foster mother, where it develops until birth. Cloning cows, pigs, mice, and even cats have since been produced using similar techniques. An egg cell is taken from an adult female sheep. The nucleus of the egg cell is removed. The donor cell is taken from the sheep's udder, this donor nucleus. The two cells are fused using electric shock. The fused cell begins to divide normally into an embryo, and the embryo is placed in the uterus of a foster mother and gives birth to a cloned lamb, genetically identical to the donor.